Hello, my name is Rhea Chatterjee, and I decided to research the molecular mechanisms that drive Alzheimer's disease. Actually, so some background about Alzheimer's disease is that it's a neurodegenerative disorder that impacts 55 million people in the world. Some symptoms include loss of memory, cognitive impairment, and difficulty completing tasks. There are genetic and environmental factors that can be shown to lead to a stronger chance of developing Alzheimer's disease. There are some treatments that exist. However, these treatments alleviate the symptoms rather than target the actual pathology of this disease. So the first theory that exists is the amyloid plaque hypothesis. This theory refers to the excessive amount of beta amyloid plaques that occur extracellularly. These plaques are usually broken down by gamma secretase, which is an enzyme that are in the brain. But in brains that are affected, these enzymes don't break down the protein, which results in the accumulation of amyloid beta, which prevents neurons from communicating with each other, which leads to cognitive impairment. So evaluating both sides of this hypothesis is very important. There are some signs in model organisms when injecting them with the amyloid beta protein, they have been shown to have cognitive decline. In addition, some immunotherapies that targeted these plaques have been shown to alleviate symptoms of Alzheimer's disease. However, there are some weaknesses of this hypothesis. Many clinical trials that have been used to use the amyloid plaque hypothesis as its basis for treatment have failed to show significant improvement in humans. In addition, some of the immunotherapies that have existed have resulted in adverse effects, such as cerebral edema. In addition, some of the measuring methods, scientists have found that FDG PET scans are a much greater indicator of Alzheimer's disease rather than the amyloid PET scans. On the other hand, there is the tau protein hypothesis. The tau proteins are responsible for maintaining the stability of microtubules in the axon and aid in cellular assembly. However, the progression of Alzheimer's occurs when there is a hyperphosphorylation of tau proteins that lead to the excessive amount of tau proteins accumulating inside the neuron, which then leads to neuronal death. So both sides of the tau protein hypothesis have very good points. Some of the strengths includes that tau PET scans have been shown to be much more accurate in predicting the future of a neuron, the neuronal death, for people especially who are in the earlier stages. And there are some immunotherapies that exist that have been using the tau's hypothesis as its basis. And in the model organisms, they found that they can replicate anti-tau antibodies and break down excess tau tangles. However, there are some weaknesses. In these antibodies, they were used in transgenic mice, but they posed as a risk in wild-type mice. In addition, the tau protein hypothesis is very difficult to study because it aggregates intracellularly as opposed to the amyloid plaque hypothesis, which aggregates extracellularly. So in conclusion, the tau protein hypothesis has a greater chance of driving Alzheimer's pathology. Alzheimer's drugs targets amyloid beta that target amyloid beta should ideally increase the cognitive skills by breaking up the plaques, but these clinical trials show that there's not enough improvement to possibly relate it to the pathology of Alzheimer's. Tau protein, although it's currently under research, it has been proven to be more accurate in predicting the molecular mechanisms that drive Alzheimer's disease.